Hello and welcome to Science Research Weekly, Season 4, Episode 12. Water from the Dawn of Space and Time. Get ready for nuclear batteries, DIY box filters, Asgardian microtubules, water from the cosmic dawn, FitQuantiles tables in SAS, teaching combinatorics using R, the 2025 Galaxy and Bioconductor Community Conference, and Python and 10 lunch break sessions. This episode covers material from March 21st to March 27th. Let's get started with some research articles. First up, we have the article, A Safe Nuclear Battery That Could Last a Lifetime. Sorry, not quite an article, but a press release from the American Chemical Society. So researchers produced a prototype battery based on beta ray emitting radiocarbon. Now, while the current design has a low efficiency, especially compared to lithium ion, I think it's only 2.86% energy conversion efficiency. A device like this, a battery like this, could run for centuries. And radiocarbon is actually really readily available. So if researchers can hone this, make it more efficient, make it more, I guess, foolproof setup, it could really be a great use for things like medicine, pacemakers that run for a lifetime, can be used for space. The sky's the limit. So really interesting stuff here. All right, next up, continuing our press release tour from the American Chemical Society, researchers presented results from a DIY, do-it-yourself air filtration system. So it's essentially just a box with a box fan on top and air filters, and then like a cardboard base, and then the fan runs, draws in air through the filter, and filters out, indoor air pollutants. So while the current design has a is rather loud, it can actually run. They ran it for like 25,000 hours, so really shows that it's efficient and robust. This was really interesting. I actually saw a study like this a couple years ago. Really like to see that they were able to install and run them to get longer term data. So cool things there. All right, we'll shift gears to our regularly programmed article reviews. First, here is long chain alkanes preserved in a Martian mudstone. So researchers were able to find, using, I think, the Mars Curiosity rover, these structures, decane, undecane, and dodecane. These are all carbon, hydrogen molecules, which provide clues on Mars's past and potential chemical biosignatures. So really interesting. This came from the mudstone. All right. Next up here, we have microtubules and Asgard archaea. So microtubules are found in eukaryotes, constructed from tubulin proteins. They're sort of the hallmark of eukaryotes. Now, tubulin homologs that don't actually form microtubules have been found in archaea and bacteria, but not true sort of microtubules. However, researchers looked at Asgard archaea, the closest relatives to eukaryotes, where eukaryotes probably came from. And they found that the Asgard tubulins appeared to actually be able to make microtubules, suggesting a pre-eukaryotic origin of microtubules. Okay. Moving along here, we have diverse ssRNA viruses associated with Karenia brevis, harmful algae blooms in Southwest Florida. So researchers were able to perform viral metagenomics on seawater from red algae, so that's Karenia brevis, during a kind of bloom or around the times of blooms. And they found a variety of viral genomes, including ones that infect bloom-forming diatoms, and also things like unclassified single-strand RNA ribovaria genomes. So really interesting to think about how viruses interact with things like algal blooms. Okay. Next, we have a very technical article here. The autonomous shaping of the piRNA sequence repertoire by competition between adjacent ping-pong amplification sites. So, Pee-wee interacting RNAs, or piRNAs, they silence transposable elements, aka jumping genes. Now, piRNAs are generated by apparently what's called one of my new favorite pathways, the ping-pong pathway. And researchers systematically compared profiles of piRNA in two related silkworm cell lines and found that sequence repertoire changed dynamically over time. So this helps us understand more how piRNAs are formed, how they persist, the levels and everything here. So a lot of technical work to go into understanding this complex system. Okay, and our final article here, abundant water from primordial supernova at cosmic dawn. So by running numerical simulations, researchers, astrophysicists were able to check with low metallicity gas clouds from the early universe. They were able to see that water probably formed in this early universe in these areas a mere 100 to 200 million years ago. So just barely after the Big Bang, cosmologically speaking. So making water likely present in the earliest of the galaxies. So that's really interesting to think how early water might have actually formed in the universe. All right, let's switch over to some research tools. First up, Rick Wicklund from the Do Loop has the quantile fit plot, comparing empirical and predicted quantities for univariate models. So he goes over the fit quantiles table from proc univariate SAS, including how to graph these values. So look at distributions, looking at these different plots. So there we go there. Two new R bloggers posts that might be of interest. First, from Keith Goldfield, a Bayesian proportional hazards model for cluster randomized trial. So this is a continuing sort of journey of Bayesian proportional hazards modeling. The latest post shows another extension, random effect accounts in clustering a cluster randomized trial. The other R bloggers post is how many ways to color this grid, teaching combinatorics using R. This is from Vidisha V. And yeah, very interesting problem, fun problem solving using R to color a 2x2 cube or 2x2 grid with colors that do not overlap. So very interesting and kind of fun. All right. Little news here. The Galaxy and Bioconductor Community Conference is going to be held in June at Cold Spring Harbor Laboratory in New York. This is the first ever joint conference between the Galaxy and Bioconductor communities. So if you're into genomics, biomedical research, environmental science, machine learning, etc., this may be for you. All right, we got a couple new R CRAN posts, packages. There's COAP, high dimensional covariant augmented overdispersion Poisson factor model. There is GG Hourglass, plot records per time of day, and GoMiner, automate the mapping between a list of genes and gene ontology categories. So with that, we'll close out here with there's nothing new on research funding. So to kind of fill in some space and lead you on to maybe some new paths, why not take some time to learn something new? I actually have a series of Python tutorials that you might be interested in. I call them Python lunch break sessions, Python and 10 lunch break sessions. There's 5 of them. You can look at the description for the video for the first one if you're interested in that. If you're watching the video, I kind of have the YouTube video up here, and it really gets started with getting Python on your computer and then the obligatory fundamentals. Cosmic thirst quenched. Thanks for joining me for Science Research Weekly. Science on.